today. I did just post the slides on Moodle for Part A, and then once we're done with that content, we'll take a break, and I'll get the other set put up there and make copies for you during break. So, and there, I also made copies of the Part A slides, so hopefully they've filtered around to everyone. So, all right, and he's also going to record the lecture today as well. So, let's go ahead and have a word of prayer so we can get started. All right, dear Father in heaven, thank you for another day of life. Thank you for bringing everyone back safely from break, and I just thank you for each one of these students. And it's a busy next couple of weeks. I pray that you'll continue to watch over each one of these students. Just give them the perseverance that they need to get through all of the exams and assignments and all that they have facing them. Help everyone to not get too stressed and overwhelmed, and just give each student the wisdom that they need. Be with us now as we have class. Help the students to be able to understand and to learn, and to be able to use the material we're discussing today. In Jesus' name.
All right. Well, so I am recording, and what I'll record is it records the screen of the tablet and the audio that goes with it, and it'll also record any drawings that I'm doing on the screen, so that you can have that as well. And then, like statistics, like Dr. Osborne, back to that exactly. Yes, I like that model as well. What we'll do is, this is of course a two-part lecture, part one and then part two, right back to back. The first part, it's a fair amount of review, but I do want to go through some of it, and mostly I want to give you an opportunity to re-solidify any of those basic genetic principles that maybe aren't quite there. I really want to encourage you to ask a lot of questions. In fact, I don't think I've taught your class before, so I believe that the fuel that powers the engine of learning truly is curiosity and questions. So I ask that you participate by asking a ton of questions, and I'll do my best to answer them best I can. All right? Well, let's dive in here. So, genetic control of cell function inheritance, I believe it's chapter 5 from your book. Starting with the very basic, first learning objective that is requested of you is just to make sure you understand the basic structure of deoxyribonucleic acid, DNA, and where it's found in body cells. So, very simply put, DNA is a very long molecule made up of repeating units. Those units are these nucleic acids, as they are frequently abbreviated A, C, G, and T, abbreviating the nucleic acid that they are appropriately named for. This sequence encodes all the information that it programs. This is lunchtime, isn't it? I see this now. I kind of get the program here. You run to lunch, grab the thing. Got 10 minutes in between classes. I got you.
We didn't even have 10 minutes this time.
Yeah, 6 minutes. Exactly. I hear you. And it's encoded in 2 strands, and those 2 strands, there's a reason they're there, and that's to allow for replication. If you didn't have those 2 strands that were together, there'd be much less effective ways of being able to replicate that molecule. It is found in the nucleus, right, of all eukaryotic cells. All eukaryotic cells that have a nucleus, that DNA is going to be found there, and it's going to house all this information. And in fact, this image that you're seeing on here is kind of illustrating, here's the normal structure of DNA, and then here's what they're calling a replication fork, where the 2 strands are being separated and then duplicated. Presumably, one of these strands will go to a new cell, and then the other strand will go to another new cell, right? Basic sort of principles. Okay, any questions so far? Okay. So, what is the relationship between DNA, genes, and chromosomes? Well, a gene is a sequence of the DNA molecule that contains instructions for building an RNA molecule. It's only about 1 to 2% of most organisms' genomes, at least when we're talking about a lot of complex organisms, is actually devoted to this. There's a lot of other DNA for other purposes, but sections of DNA will actually have instructions for creating an RNA molecule. And that RNA molecule, what's going to happen to it is it's going to come out of the nucleus, be translated into protein, and then that protein is going to carry out a particular job. Each of those proteins is like a little molecular machine. That's the best way of thinking about them. Every protein, your entire cell, is made up of lots of little molecular machines, all going about doing their business. And each of those machines has to come from somewhere. And where does it come from? It comes from the messenger RNA that was translated, and that messenger RNA came from the genes that are encoded within the DNA in the nucleus. Questions on that? I know it's pretty basic. I'm sorry. I apologize. We'll get to some good stuff, but I do want to make sure if you have questions, do ask them. Even if they're just curiosity. Happy to entertain a few of those. Codon. So what's a codon? Just be aware that when messenger RNA is created from the gene, from that DNA copy, the messenger RNA, it encodes amino acid sequences in sets of 3 nucleotides in a row. So it might, I'm just making some up now, and I don't know my code anymore. I used to actually know it well enough. We did a lot of genetic engineering in the lab. We would mutate viruses in order to treat diseases, and could tell you a lot of what the codons were, but I haven't used it in probably 7-8 years now, and so when you don't use it, you lose it. But nonetheless, each of these codons will encode for a particular amino acid, and as the ribosome, which can read messenger RNA and translate it into protein, comes along, it'll take each of those codons and translate it into an amino acid sequence. Okay? And I may even have no, not sure if I have a picture of that or not. That's okay. Any questions?
Sequence of things.
The amino acid sequence? The amino acids are linked together to make the protein. So the protein, in fact, I definitely need an image because I'd like to, there's messenger RNA being produced. That's good. Some splicing happening. That's great. Sorry. See if I can pull together an image of the translation, what you're asking. I don't actually have a good image to show you what you're asking there, but what happens is, yes, the amino acids, each of them is just linked together to form a larger sequence of amino acids, and those fold in to make a single protein. And each protein, its shape is dependent on which particular amino acids are put in there, and the shape of that particular protein determines its function. Structure dictates the function of it. And so to make these little molecular machines, we take the DNA, which actually this shows that image pretty good right there, actually. Take the DNA, you make a copy of it into messenger RNA, and that copy is pretty much identical. The only difference might be that you have the uracils, the U's instead of the T's, but other than that, it looks very, very similar. That messenger RNA will leave the nucleus and go out into the cytoplasm, and then from there, it's read by a ribosome, and for every 3 of these nucleotides, a single amino acid is placed in, and depending on the sequence of this, that determines which amino acids put in there. Does that help at all? Okay, fair enough. Okay. And that's essentially the central dogma of all molecular biology. That's it, right there, is that DNA is transcribed into messenger RNA, which is translated into protein. There's about 5 Nobel Prizes right there that's at the center from the 1950s and 1960s, understanding how biology works, right there. That makes a lot of sense. And it goes across tons and tons of different kinds of organisms, not just humans or not just mammals, but virtually every organism uses that basic fundamental set of principles. Bacteria and things do it sometimes a little bit differently. They don't have a nucleus and whatnot, but still, it's still very similar. Okay. I'm not going to go too much into processing RNA because I don't think that's actually part of your learning objectives. Unless I'm mistaken, exons and introns are interesting. Splicing does matter a lot. The disorder I worked on for several years was actually a splicing disease, and you can get messed up in this. Just like any part of the biological system, you can have errors in it that can lead to diseases. But in this context, I don't think there's anything that actually goes over splicing, so I'm not going to linger over that for too long. Here's just more information on translation. And again, these are just kind of those basic principles. The amino acids are brought in by transfer RNA, the tRNA, and all those things are just little adapters to link a single amino acid to a specific sequence of the codon there to make sure that it fits the right sequence. And it's just facilitating bringing amino acids to the messenger RNA and then allowing those amino acids to be linked together by the ribosome. Okay, so a little questions here. We have several questions that we get to answer together as we go throughout the lecture today. Which type of RNA, we've got several different kinds of RNA, but which type of RNA moves from the nucleus into the cytoplasm and it carries the genetic code? Definitely A, right? Messenger, right? It's a message between the nucleus to the cytoplasm, allowing it to carry that message to the ribosome and be translated. Okay. Now, this is tricky. This will be an interesting question, perhaps. See how you interpret it. Let's rank these problems as best we can from least serious to most serious is how it's asking. It's from least serious to most serious. So kind of let's say, 1 least serious and the most serious is like 3. So let's see what we can do here in linking these up. Okay, a serious error in the messenger RNA processing for a gene. Okay. A serious mutation in the gene for the transfer RNA with the anticodon AAA. A serious mutation in the gene for ribosomal RNA. So what do you guys think? So let's give me some ideas, like one at a time. It's like, think of one of these. Which do you think? Should we start with the least serious? Maybe the least serious. You think least serious is this first one here? Okay, so you're going to have one gene in the genome. By the way, we've got about 20,000-ish genes in the human genome. Okay, so one gene in the genome, it's got a serious mutation. Well, that could still kill you. I mean, that could still make it so that it's incompatible with life, but maybe it'd be a little bit less serious than some of these other ones. Here's another one, a serious mutation in the gene for the transfer RNA with the anticodon AAA. What do you think about that? Sounds like more serious than a single gene. Okay. Let's rank that one versus the last one, a serious mutation in the gene for ribosomal RNA. That one would probably mess you up good, right? Because you can't translate anything. None of your genes would ever get translated into the molecular machines. Many of us have mutations, or many individuals can have a mutation in a single gene and maybe have just a disease. If you have a serious mutation in ribosomal RNA, you might not be able to make any of your proteins, and that's likely incompatible with life, if that makes sense. In other words, if that mutation occurred, it would arrest development of that developing zygote. And then same thing with the tRNA. This one would mess up every single protein that had, or every single gene that had a encoding this codon. That means that there'd be a mutation in every protein product. Okay. Any other questions on follow-up of that? Oh, sorry. So that would be from top to bottom there. So this is the least serious. This is the medium serious. And I would definitely say that'd probably be the most serious. Although, again, with a caveat, there are a number of even single gene mutations which would be flat out incompatible with life. So those can be pretty bad too. But just as a general rule of thumb, that's how you probably would want to rank those. Okay. So gene expression is the protein produced. There's not a lot in your learning objectives, I don't believe, about this, so I'm not going to spend too much time. But basically, not all of your genes are turned on at the same time, and they come and go. Some are developmental; they're only active at certain times of development. Sometimes they're active only for certain periods of the day and then go off. And genes basically can be induced to produce messenger RNA and subsequently produce a protein, or they can be repressed. And there's molecular machines that are out there that are helping to control that. So here's true or false. If a gene is induced where it should be repressed, the result can be injury to the tissue. Of course, I haven't given you information maybe to answer that. But the answer is definitely true. There is, like for example, here's a good one. If let's say a gene responsible for a digestive enzyme which is maybe only supposed to be expressed in an inactive form in the pancreas, suddenly were to be induced in the lung, there could be significant digestion of the lung tissue, for example, right? And there'd be a real problem because we turned a gene on in the wrong place. Really, to be honest, the entire set of diseases of cancer could really be linked to this. Genes that normally should not be on and not allowing cells to divide are turned on. And mechanisms, some genes that are supposed to be stopping the cell cycle are turned off. And so cancer as an entire disease, in the broad sense, is a whole bunch of genes, and different ones in different cancers, that are either on or off inappropriately. All right, so moving right along. Genes are found on chromosomes. That's how we're organized. Your DNA is about 3 billion base pair, about 3 billion base pair, but it's not one long strand. It's chopped up into chunks, okay? And those chunks are called chromosomes, and they're numbered very simply. 1 is the longest chromosome, and then all the way down to 22 is the shortest chromosome, and then they separate our 23rd pair, because the 23rd pair, of course, is the sex chromosomes, X and Y. And depending on the sex of the individual, they either have, of course, 2 X's if they're a female and an XY if they're a male. All of the chromosomes 1 through 22, they're considered autosomal because they're not the sex chromosomes. And the sex chromosomes, of course, are just X and Y. Okay, so we'll talk about autosomal recessive inheritance. When we say autosomal, what you should immediately say is, well, I don't know which chromosome, but I know one of the first 22 chromosomes is involved in this disease. Does that make sense? And then, of course, if we said it was an X-linked disorder where it's very specifically laying out, okay, well, it's going to be linked somehow to the X chromosome, one of the sex chromosomes. So be able to define that term autosomal. Okay. You've also probably gone over meiosis and mitosis, right? So hopefully this is a good review thing for you. Which one of these is related to the production of eggs or sperm, also known as gametes? The meiosis, right? And what's unique? Tell me that. What is unique about meiosis that's kind of really different than mitosis? There's lots of different things, but tell me some different things. What's that?
Oh, I was just saying it creates cells, like in humans it creates cells with 23 chromosomes instead.
Right, instead of having a full complement, where we normally have a full complement from our mother, full complement from our father, 46 chromosomes, and it's really 23 pairs of chromosomes, these gametes only get one set of that's going to be passed on. That makes sense. Good. What else is in there? Yeah, there's more steps, kind of. Yeah, you could the way that it's completed. There's kind of meiosis 1 and then meiosis 2. It takes a little bit more time to separate that information out because it is being separated more. Do you notice the colors there? Do you see that coloring? So, like at the very top here, where it's starting, you notice they're just showing one chromosome. For example, this might be chromosome number 1, and you notice there's a blue and then there's a red, right? Well, the blue might be that it was inherited from your father, and the red might be from your mother, right? But you still have full sequence of all the genes on chromosome 1 are present there. You basically have 2 copies of it. And then notice what happens to the colors over time, right? They line up, and there's actually mixing between the chromosomes. So the end result is the gametes have chromosomes that are not purely from your mother or not purely from your father, but the chromosomes will actually be hybrid chromosomes. Part of that chromosome might be from your mother, and part of it might be from your father, right? Homologous recombination, I'm not sure that rings a bell for you. It's an important step in increasing the genetic diversity of the population so they can really get lots of variations, right, in terms of offspring from a pair. And then mitosis is much, perhaps in some ways, more straightforward. It's not on this screen here. Let's see if it's on the next one. It is simply the straight-up duplication of DNA, and both daughter cells get a full complement of 46 chromosomes. Does that make sense? Okay. All right. And that's what this is listing. Meiosis is being described there. Oh, and then here we go. We've already answered this. Not mitosis, but meiosis results in the formation of gametes. Okay. Okay, I do have a little image of crossing over here. That's kind of important. We're going to talk about chromosomal disorders, and perhaps the one thing that I would point you to here is that many of the chromosomal, I wouldn't even say most, but many of the chromosomal disorders are linked to problems with the crossing over process. Imagine if this somehow got messed up where you didn't have a perfect switching of DNA here, but maybe it's unequal, right? Maybe one chromosome gets a little bit more DNA, and then the other one gets a little bit less. Ooh, might end up having not fully 2 copies of every gene then in the subsequent, zygote that's produced from that. And that we'll see that it's disorders of crossing over that can often cause that. Okay. More basic information here that we'll be using a lot, and that's genotype and phenotype. So genotype is just someone's genetic material, and phenotype is someone's physical characteristics, what they actually look like. So let's do some example. Actually, here's a good discussion question. I'll just use this. If 2 people's genotypes are different, will their phenotypes be different also? Kind of tricky one, isn't it? Explain different answers. So I heard some people say, well, yes, and why would you initially say maybe yes on this question? How could you defend that side of it?
Because my genes are different than her genes.
That's right, because the genes produce the proteins, and if the proteins are of different shape or size, that can result in a different phenotype, right? If my genes encode a mutation that make it so I'm lactose intolerant, but yours do not, then obviously there's a different phenotype between the 2 of us there. However, phenotypes are not always just produced by a single genotype. Does that make sense? And so that could be that, more than one person can have blonde hair, but it may not be they have the exact same genes that are encoding that blonde hair. It could be different genes, actually. I'll give you another example. Over 200 genes that we know of so far control height, right? As well as, of course, environmental factors. If you don't get enough nutrition, your height will be stunted and all that. But genetically wise, about 200 genes, and each of those 200 genes, a little bit of that gene contributes to height. So you could have 2 very tall people who, out of those 200 genes, could have the tall versions of different parts of the different genes out of that 200 that produce the height. They could be the exact same height, but it's different genes that's producing that height. Okay? So this is a trick question, I think. I mean, it could go either way on that. Okay. Oh, goodness. I think we're going to start talking about Mendelian genetics, but we haven't yet. This is interesting. What's that?
You can change your order.
Oh, sorry. Yes. I apologize, guys. This is actually the first time we radically changed the course over the summer in terms of the learning objectives, making them a little bit sharper, and then the slides as well. What this question was going after is, we'll study in a second here, Mendelian genetics. There's dominant alleles and recessive alleles, typically illustrated by capital letters for dominant traits and lowercase letters for recessive traits. And here, BB represents a genotype, and basically they're saying no, not necessarily this links up to a particular, they're listing the genotype of the individual, and yeah, don't even worry about it, sorry. That basically all they're doing is just showing that they're listing a genotype there instead of a phenotype. Phenotype is always what we actually see on the outside, or a characteristic that we can measure. Well, here we go. Gene interactions. I just illustrated this about 10 seconds ago with polygenetic. Polygenetic is a very fancy word. It means one thing, and that is there's lots of things, both normal characteristics and diseases, that are polygenic, meaning that many, poly, genes, genic, many genes influence that trait. Okay? So let's think of some examples. We know that there are specific populations that are at higher genetic risk for heart disease, for example. But it's usually many, many genes, not just like a single gene, but many genes that actually influence that particular outcome. Okay? I gave you the height example. That's a really good example of several hundred genes that affect a single. Here's another good one that's just kind of fun to think about. There is two versions of an actin gene. If you remember back to your muscle physiology, actin and myosin interact and they produce force within muscle cells. And you probably are also aware of fast twitch and slow twitch muscle cells, in which slow twitch can produce significant amounts of force, but they don't do it quite as fast. And fast twitch, of course, by their name, they twitch a lot faster. They can produce a lot of force, but they're not in it for the long haul. They tend to use up their energy very quickly. So sprinters ideally have lots of fast twitch muscles, and then long distance runners have more of that slow twitch muscles. The actin gene has been well studied, one of the actin genes, there's actually many of them, and there are different versions that will bias the person to have more or less fast or slow twitch, depending on your genotype. And it's been best studied in Olympic athletes, where they've studied, and sure enough, in Olympic athletes that are in the sprinting competition, there's a higher percentage of them that have the mutation or the genetic variance that biases their muscles genetically towards having lots of fast twitch muscles. And then, not surprisingly, in the marathon runners at the Olympic level, there's a greater percentage of them that genetically have an actin gene that biases their muscles towards the slow twitch. Not all of them, because that's not the be-all, end-all of being performance, but it does help. So yeah.
Doesn't that kind of follow the saying that like the sport actually chooses the athlete?
Yeah, it would actually a little bit, especially at the highest level.
You
right?
only have a certain number of these types of muscles, so you're likely to be better at this because you have this certain amount of like fast twitch
That's
muscles,
right.
as opposed to other people.
That's right. I would never discourage anyone from starting their kid off in, pee-wee running or baseball or whatever because they think, oh, you got slow twitch muscles, you're forever doomed to ever. No. But at the very highest levels of competition where tiny things make a big difference, yeah, you may not get to be at the Olympic level unless you perhaps have a genotype that really would match that. Makes sense. Now, something else is, and these are also examples that we can point to from what we just talked about, often the phenotype, our traits, are highly impacted by both genes and the environment, the whole nature versus nurture argument, right? So in terms of height, I mentioned if you don't get enough nutrition, you will never reach your potential genetic height, right? And so there's nature is influencing it based on the environment. Nurture, inherit also affects it. Epistasis, this is where one gene can mask the effect of another. This would be, I keep going back to the height one because we've already talked about it, but this would be in the case of, you can actually have, because there's 200 genes for height, you can actually have a whole bunch of those genes which are the short allele, but still be a very tall individual if the other genes that you have out of that 200 complement, if you have a whole bunch of them that are tall that you have a tall version of that gene, if that makes sense. And then the tall ones would mask the other. Some genes are complementary in that they very much depend on each other. This is because the proteins they're producing are molecular machines that often are involved in multi-step processes. You think about some of the processes you've been exposed to, like the glycolysis cycle or something like that. To be able to make that cycle work, it requires many genes, many proteins to be able to actually get it working. And even if there's something only wrong in maybe one particular gene, it could actually be messing up something for an entire set of genes and getting their job done. Okay? And then two genes often work together to perhaps create a new phenotype, and they can work together, and that's collaborative. All right. Any questions about anything there? I've already started using this word, and I apologize, but now we get to define it. Alleles. Alleles are copies. Copies is an okay word. I like the word version. A version. Yeah, version of a gene. So, you might have a single gene. Let's choose a really standard one that you might have, like hemoglobin. That's a perfect example. You might have hemoglobin, and there's actually multiple parts of hemoglobins, like beta chains or something. And the genes that encode for it, there can be different versions between different copies of your gene. So you know that every person has 2 copies of a gene. So I'm going to write here, here's one copy, here's another copy. Didn't mean to make them actually one longer than the other, that's okay. But you might actually have one version of the gene be only just slightly different than the other, in that there maybe is like a single nucleotide difference, like one nucleotide difference. And in the case of hemoglobin, you can think of like sickle cell anemia, where on the beta chain there's just one amino acid, one nucleotide that's different, which results in a single amino acid change. And if you had one of those in your genome, that could be on, for example, maybe maternally inherited, and then one from paternal, you would have 2 different alleles for the same gene. Does that make sense? 2 different alleles, 2 different versions of the same gene. The overall gene structure would be pretty much the same. So the overall, if you actually studied it, it'd be A, C, T, A, whatever. I'm just making that particular up. And this might be one version, very oversimplified. And then the other one, very often, would be, very, very similar, but maybe instead of a C there, it had a G. This would be considered one allele. This would be considered another allele. And this helps produce some of the variation you see in the population, both non-disease causing and disease causing. So alleles are different versions of a gene, and you have potentially 2 alleles for every single gene that you have. Now they might be the exact same, right? You could have inherited the exact same sequence from your mother and father in terms of they're identical, or they could be a little bit different. Questions on that? Okay, I think that hopefully that makes sense. So that then leads to these terms, homozygous. That means that the 2 alleles are the same. They're identical. And then heterozygous is the separate term there, and that means you have 2 different alleles. Okay. Now coming back to these terms, recessive and dominant, we talked about those before. The very simplest definition, this was all done by Mendel back 1800s, studying pea plants, is that dominant traits, if you have a single copy of that dominant trait, it will produce the phenotype. Dominant traits produce a phenotype, whereas recessive traits, you need to have 2 copies of it. So if I have little a and big A, I would have the big A phenotype. So let's just make this up and say it would be, let me give you an example. Let's do a real example. Let's do Huntington's disease. Let's do that. That's a dominant inherited disorder. So Huntingtin is the gene that encodes for Huntington's disease. And for example, if you have 1 copy of the bad Huntington's gene, which we'll do as capital H here, and then 1 good copy, you might say, 1 good allele, you still get Huntington's disease. To not have the disease, you need to have 2 of the good copies, which are recessive. And so you'd have to be homozygous for those, which most people are. There's not, the rate of Huntington's is 1 in 10,000 or roughly around there that people are carrying it.
Frankie,
Yes?
they're not going to know what that disease is. You want to just...
Oh yeah, Huntington's disease is it's a progressive neuropathy that causes breakdown of lots of neurons in the brain, which result in a phenotype of both chorea, which is uncontrolled movements that are that appear somewhat coordinated, but they're not intentional. So they have unintentional spastic movement, and then also a deterioration of mental function. Both of those are progressive in terms of loss of voluntary control over your motor function and loss of mental capacity. And unfortunately, it is a lethal disease that usually has a varying onset, but takes a few years before it becomes, that results in death. Does that help? A very positive. Oh, wow. Okay, now we're so.
And it's a dominant,
Yes.
so it's easily.
Yeah, we'll get to the inheritance pattern in the next lecture, but absolutely. It means that 50% of the children of a person who has that allele, they have a 50% chance of inheriting that allele. And if they inherit it, that means they will have that particular disease. Unfortunately, still no treatment for this that's effective.
At what age is that usually expressed?
It's at different ages. So it starts, it usually starts in a family. Someone will get it in their 60s, and then they'll pass it on, and the next person will get it in their 40s, and the next person will get it in their 20s, and eventually the next generation will get it when they're really tiny, like they're 4 or 5 years old, and then it burns itself out because they weren't of reproductive age. It's called anticipation. The gene is getting longer between generations. There's this repeating section of the gene, and every time the DNA polymerase goes across that repeating section in producing the gametes between generations, it slips and it copies extra amounts of that repeating segment because it's getting lost in it. And those repeating segments, the longer they are, the faster the symptoms come and the worse the symptoms are. And so it's terrible for families, right? Because it comes on and just burns through a family tree.
So for a person who's not expressed till like 60, is that because the telomere shorten? Or
the reason it's not expressed till they're 60 years old is because the repeats that are within that gene, they're just not that long yet. They're long enough to cause symptoms and to cause the disease, but they're not long enough that they're coming on sooner, if that makes sense. The magnitude of the disease is proportional to the... It's probably as much more than you need to know for the test, but that's maybe interesting to know. Does that help? Okay. So that's okay. So heterozygous, homozygous, recessive, and dominant. Any questions on any of those? So let's switch over here. Let's say there's recessive traits, and let's say you have a recessive trait, and you're heterozygous. You could be a carrier. This is a good. I'll give you another good example of a disease. The disease I spent most of my time working on was spinal muscular atrophy, which is a disease very similar to Lou Gehrig's disease. You probably heard of Lou Gehrig's, ALS, amyotrophic lateral sclerosis. Those diseases, both ALS and spinal muscular atrophy, are a progressive loss of motor neurons in the spinal cord that result in paralysis of voluntary. In that disease, it's a recessive disease in terms you need 2 bad copies. Right, you need 2 bad copies, and so the gene that causes it is SMN, and so you would need, most people have homozygous, these are capital S's, of the healthy allele, the dominant allele, but then to actually have the disease, I shouldn't use S, let's use something else, I'll use the letter N. So normal, that's what most people are. But about 1 in 40 people, so there's a decent chance someone in this room is a carrier, about 1 in 40 people carry a recessive allele. And this recessive allele, it doesn't work. That version of the gene, whenever it's made, it just doesn't do its job. So you're okay, actually, if you have 1 good copy of it, because the good copy continues to do its job, and it's enough to cover the base, and you have enough to get by, and your motor neurons survive, and you never even know it. Go through your life, you could be a great athlete, you could be whatever, you never would know it. But when 2 individuals that are both carriers, right, have children, there's a chance the children could end up with the disease-causing genotype, which would be the homozygous recessive allele. So carrier is the term we would use for a person who has 1 dominant and 1 recessive allele. Yes?
This is a question out of curiosity, so
Yeah.
if you don't know the answer,
That's okay,
I
yeah.
really think. But is it more like is the dominant diseases or
Yeah,
ones less prevalent because they do, as you said, tend to like burn themselves out?
Yeah.
Versus the recessive ones.
Yes.
There's they can. Have
There's
these,
selection
like,
pressure
carriers.
against having a lot of dominant disorders. You betcha.
So it can be that.
Yeah, exactly right.
Changes are more common.
It's almost like you can imagine it being, there's different viral diseases. Many of us know, like, for example, rhinoviruses that cause the common cold. They are so prevalent everywhere because they don't kill the host, right? And the host can walk around and spread it everywhere else. But then there's diseases that are very incredibly aggressive, like hemorrhagic fevers, where they'll kill you in just 2 or 3 days. Well, much harder for that virus to spread around because it's so intense, and so same thing with the dominant ones. Now, not all dominant ones are terrible. Some dominant alleles don't hardly cause any problems at all. And of course, dominant recessive don't even have to apply. Any other questions on any of these terms that we've gone over here? Very rarely, and this does happen, you could be hemizygous, where you only have one copy of a gene. That actually happens with spinal muscular atrophy. Going back to the homologous recombination, the crossover, sometimes when that lines up, it'll actually loop out a part of the DNA and just delete it. And that means the resulting chromosome is missing some genetic information, and so you might get one normal copy from your mother, and then you don't even get a copy of the gene from your father, and that would be a hemizygous person, that they only have one copy of the gene. They just literally don't even have another copy of the gene. They have one. Yeah. Well, it depends on what the situation is. If it's a gene that one copy, if you have one functioning copy, you're okay, then you don't have disease at all. You might just be basically carrier for potentially, passing something on. If it's one where you need 2 functional copies, it could cause a phenotype that could be related to that disease. So, question?
So I know, like, in basic classes about this, like
Right.
biology in high school or whatever,
Yeah.
they talk about like eye color as just being like, oh, brown is, dominant, so it has
Yeah.
this. But
It's about 30,
isn't it-
about 30 genes,
Yeah,
roughly.
it's more complicated than that. So
Right.
what are the kind of combinations that add for like hazel eyes or green
Yeah.
eyes or those ones that we don't really talk about?
So that's not my PhD, so I'm not sure if I can give you the details, like the inner workings of that. But I do know it's roughly in order of about 30 genes that influence eye color. And so my imagining is that it's likely dependent on some of those genes, on which ones are expressed and which alleles someone has for those particular genes and which alleles they don't have. There's actually a pretty fun little program out there that some scientists made where, taking into consideration all 30 genes, you can upload your genotype. Maybe we'll talk about this later too. There's direct-to-consumer genetic testing now, like 23andMe and other companies that do, like, you spend $100 or $200 and then send them in a saliva sample, and then careful, whoa, easy now. I can see ordering a kit in the back there. Like, whoa, we'll talk about why that you should be fully aware of implications if you decide to do genetic testing. It's an interesting thing. I have done genetic testing myself, but be aware of the implications for genetic privacy and be able to say that that's a risk that you're okay with. Anyhow, there's some scientists that once you get that information out, you can kick in your genotype and it predicts eye color, and so it'll show you an eye. And it's like, okay, is this your eye? And then it's one of these self-correcting mechanisms where people who've done their genotype can report in and compare and say, oh, it's off a little bit, and then they try to improve their algorithm to predict it based upon the genotype of the individuals. All right, almost through this one. So the Punnett square is just to show, of course, we get gametes from mother and father, sperm from father and an egg from mother. And here's the key thing, and that is you only inherit, one copy of each gene from your father and one copy from your mother. But recall that, of course, the mother and father have this diploid genome where they do have 2 copies. It doesn't have to be heterozygous like this. It could be, capital D, or lowercase d, lowercase d. It could be either one. But regardless, you can actually then lay out and predict what's the relative proportions you would expect of offspring from that couple. Does that make sense? This comes back from Mendel, yeah.
Is this what they do with genetic counseling?
That is exactly what they do with
Just
genetic
like for
counseling.
all the diseases?
Right. Now, unfortunately, Mendel did some wonderful work, but it only applies to the Mendelian genes. And unfortunately, there's a lot of genes that really don't fall into his category. But nonetheless, Mendelian traits, Yes, this is the sort of stuff that we absolutely want to do. So, in this situation, let's pretend as though we have a recessive disease here. Let's say that small d is a recessive disease. Neither the mother or the father carry the trait. Right? They don't express the trait because they have one good copy, but they each can pass on a bad copy. And so what rate would we expect to see here? Well, we would expect to see 1/4 of their offspring would actually inherit 2 good copies. In fact, they wouldn't even carry the disease at all. 1/4 would actually have the disease, right? They'd have 2 recessive copies of the allele, and sure enough, they would have the disease would be expressed. And then half, if you add up this group and then add up this group together, half of the predicted offspring would be carriers of that particular allele. Does that make sense? And the way we're just doing it is you're just taking down, take one letter down to here, and then take the other letter down to here, and then just do every possible combination. Take this letter over here, and then this letter to here, and you just do every combination. That gives you your genotypes. Okay. Yeah, linked genes. This just means these genes are close together on the chromosomes, and this was really big in the days we were doing a lot of genetic mapping to map where genes are that cause certain diseases, but I'm not sure if, I don't think there's a learning objective on that. I'm not going to go deep on. That's a fun thing to think about. Wasn't that long ago where we had no idea where a lot of the genetic diseases, what gene was causing them, and so you had to do a lot of careful sequencing and studies to figure out what genetic sequence in this person is always correlating with the disease, and story for another day. So recombinant DNA. This is big. This is where you can take DNA in a test tube. This is what I did for a living for a while, or not a living, it was for my research. And you can, in a tube, take DNA and combine it and pretty much make whatever you can imagine. In other words, you can take any sequence of DNA and recombine it. You can take a jellyfish gene that expresses makes a fluorescent protein, and you can put that directly into a cell that eventually you can turn to a fruit fly. You can, goodness, what we did was we genetically engineered mice to replicate the disease. Mice don't get spinal muscular atrophy. There's no mice anywhere that get it, but we made mice that would get it. And what we did was we genetically engineered, they have a similar gene to humans, but they only had normal copies. We took the human bad version and essentially put that into the mouse. And so the mouse, I know it's terrible in one sense. It's not, didn't do it just for fun of let's just do this for fun. No, it's because in the effort to try to find treatment in clinical trials, it's really unethical to try out all kinds of prospective therapies on directly onto patients without having some idea that there's a good chance there might be some good that could come out of it and not a lot of toxicity. And so the mouse model, of course, was there for evaluating treatments. But the way what your book and whatnot is illustrating is this can be used in the lab to make all kinds of things that are very helpful to clinical medicine. So, for example, you can make synthetic insulin. You can make synthetic EPO, or erythropoietin. The blood factors, the clotting factors, right? Like factor VIII. Tissue TPA, so trying to block, go after clots, right? To help break down clots, clot-busting drugs. All of these are normal things that are produced inside of our bodies, but can also be synthetically produced in the lab, so that we can use them in the clinical setting. And it's because we can chop up DNA in a tube and recombine it in a way that you want to recombine it, and then put it in potentially a new organism to change that organism. And usually these things are made either in, you usually can make them in bacteria. Sometimes they'll make them in cell culture, where they just have cells that are literally sitting in a dish, but they always have some sort of other organism that's producing it. All right. Oh, that's interesting. True or false? I haven't said this, but let's see if we can answer it together. Recombinant insulin, when it's produced by gene isolation and cloning, the bacterial gene for insulin is expressed to make insulin protein. What do you think about that? Do bacteria need insulin? No, we need insulin to be able to control our blood glucose levels. Bacteria literally just try to, grab the glucose and other nutrients out of the environment, and they have no need for insulin whatsoever. So they don't even have an insulin gene, so absolutely false. The insulin is coming from the human side. We copy it from the human chromosomes. You can insert it into basically some bacterial DNA and put it in the bacterial cells. All right. Let me just go over a few of these. I think this is our last slide, and we'll take a break after this slide. Does that sound good? Let's do that. All right. So first one, these are some other learning objectives I'm just going to comment on. Discuss how new technology such as gene therapy is changing disease prevention and treatment. Yeah, CRISPR-Cas9, that is huge. This actually has revolutionized the field of gene therapy. So gene therapy is any treatment that is fundamentally based on genes. Now, what does that mean? Let me give you an example. The disease that I worked on, spinal muscular atrophy, basically every single one of those patients is missing a good copy of the gene. It's a recessive disease. They got 2 bad copies. They just need 1 good copy. That's all they need. So one theoretical treatment is you take that gene and you somehow get it into the patient. But it's kind of hard to get DNA inside of cells, and so usually you have to use some sort of a mechanism or vector. So most often viruses are used. Take a virus that normally can get inside cells very effectively. The outside of the virus is actually designed to attach to the surface of cells and get itself inside. Many viruses have mechanisms of even getting their own nucleic acid into the nucleus. A lot of gene therapies out there will use a virus to be able to deliver the missing genetic information that's needed by patients. CRISPR-Cas9 is huge in that, there's a little link there that you can watch on your own. It basically is a bacterial system that has been discovered in the last, I guess it's been about 8 years now, that's completely revolutionized how we do gene editing. And up until this point, it's been very inefficient to actually directly edit genes inside of living organisms. It's just really hard. Even if you get the information in there, it often doesn't actually, you can't actually make that genetic information be incorporated into the host DNA. Well, CRISPR-Cas9 is a beautiful mechanism that bacteria have had that actually protects them from viruses, but that same system can be harnessed and changed so that we can take any sequence of DNA and potentially put that into an organism. And it's much more efficient than anything ever in the past, and will be pushing into the future as to making a lot more genetic diseases treatable. Okay. Low-cost, high-speed genomic sequencing. This is unbelievable. I could talk about this for an hour. Yeah, question?
Okay, so when you put a gene into a human body, is that kind of like blood transfusion process? Will the receiver, whatever, attack the gene that come in?
Yeah, often the immune system will try to attack the virus, but in most of the situations what happens is the person's inoculated with just billions and billions of copies of the virus that the immune system cannot capture all of them, and many of those viruses will end up hitting target cells and getting inside of them and delivering the DNA. But yes, your immune system would respond to it. They often use viruses that are relatively innocuous, like adeno-associated virus. 60% of the population is currently infected with adeno-associated virus, so it doesn't cause disease. It's we always talk about viruses, causing disease and all. These replicate so slowly that they really don't even cause any disease. They're just like a little parasite. Like on whales, there's parasites and all this stuff. Well, we have parasites too, and they're viruses, and often those can be harnessed to do something, and there's not predicted to be much of a disease that then would occur. And also they're crippled. They're crippled viruses. They can't really replicate themselves. Yes, they're crippled. I know, it's a horrible way of saying that, but it's true. It's true. They are crippled. Low-cost, high-speed sequencing. Since 2002, the cost of sequencing has gone down so fast, and the speed of it has risen so fast. It is much faster than microprocessors have changed. So you haven't quite lived long. Well, maybe you have. You can think about the most elementary computers that you worked with whenever you were as small as you were, like 5 or 6 years old. Go back, think about the computers, what they looked like then, and what they could do. right? Of course, way before iPhones, right? Go back before that. They were very simple and they couldn't do that much, but boy, they're much faster. They can do a lot now, right? Genomic sequencing has outpaced in technology microprocessor transformation by far in the last 10 years. What does that mean? It means that it used to cost us $3 billion to do a genome and several years as of 2002. In 2007, the next human genome that was done cost about $10 million, took about 6 months to do. Fast forward to 2010, it cost about $10,000, took about a week. Fast forward to now, it takes just a little less than a day, costs less than $1,000. So that really has revolutionized genetic testing. It used to cost $3,000 to go just check one gene, like the BRCA1 and 2 genes for breast cancer. One gene, $3,000 to $4,000. Well, now we can sequence the entire genome for less than $1,000. So it makes a big difference in terms of clinical diagnostics and also the impact it's going to have on our just society in general. Whole Gattaca, you guys know about Gattaca or not, that movie, but basically it envisions the future of like what would it look like if we had this genetic information available everywhere and that we were using it to select embryos and we were using it to select people for jobs and we were using it to, on down the line. BRCA1 and 2 are the breast cancer genes, and sure enough, especially after Angelina Jolie had her prophylactic treatment to prevent cancer, has gotten very much recognized in the media. And these genes do, if you have bad versions of these genes, bad alleles, dramatically increase the risk of cancer. Some of the alleles have a 80-90% chance of causing cancer before the age of 70. So really high risk of having cancer. Okay. And then lastly, genetic counselor. Genetic counselor, this was mentioned earlier, it's a great field. Nurses can do genetic counseling. I don't know if you knew that or not. There's a nursing genetic counseling emphasis that's out there. It's usually associated with a master's degree. I don't know a ton of the details about it, but there's that. There's master's degrees which are specifically tailored to being just genetic counseling. And what do all these individuals do? They sit down with patients and they talk to them about, okay, here's your risk for potentially passing on to either your children, or here is the risk that your family members may be at because you have a genetic particular condition. And they are involved both in the pre-decision about should we do testing, and then also when results come back, here's what those results mean. And they do lots of patient teaching. So they're really into genetics and into explaining things to patients. And that wraps up the first section there. We'll take a break for maybe, 10 minutes, Ellen? And then I think Ellen's going to print off the new PowerPoint for the next section, upload the next one. We'll come back in 10 minutes. We'll do the next part.